This is, hold on, let me put on my AirPods. I think hopefully this will make the audio a little better. I'm Adam Fields. I am in my office, but you can't tell because I've got this really cool background. This is in La Jolla. I used to live right here for about 10 years, just right there on the coast, about 6 miles south of here. And living by this beach really helped me get through my postdoc. So, enough about that. What I'm here to do is tell you what we do in the lab. I'm a professor, associate professor at University of California, San Diego, and I study the brain. I'm going to tell you a little bit more about that. But the reason I'm doing this is because taxpayer money funds my lab, and it funds most science. And right now, there's a lot of questions about what taxpayer money is going to, especially when through the federal government. And so we're having to defend that. But good news is we should be telling you anyway. So although I've only had one big grant, I work on other grants too, and trying to get more, and hopefully I'll have a long career. So why not tell you what's going on? I think it's a good practice, and I'm going to try to do better at it. And I'll obviously not be great at it at first because I'm not used to speaking into a camera, but we'll get this going and I'll just post it. It's going to be unedited because I don't know how to edit these things. And so my whole point is to tell you a little bit about me, where I come from, how I got into science, what we do in the lab, who works with me. I'll tell you a little bit about UCSD. You can tell me what you want to know about the lab, about academic science, how we try to get into the corporate world as far as turning our research into products and into things that will help society, which is really the ultimate goal, to try to make people healthier and figure out things and ways to make people healthier, things that people can do or that doctors can do to treat people. In our lab, we study the brain. So we are technically, I like to call it a translational neuroscience lab. Sometimes I'll say experimental neuropathology lab. So I will explain a little bit about what that means. I don't want this one to be too long because these things can drag on forever. But if I try to talk to you once a week and just put something out there, then I think we can cover different aspects of the lab and what we do and how we get grants. Um, I don't know, whatever you want to know. We have quite a few projects, so I'll just be telling you about maybe one project a week, problems we have in the lab, things, cool equipment we have in the lab. All right, so I thought I would start. I'll show you a our lab website. This is not, I don't give it enough attention, but maybe this will spur me to do so. So what I'll do is I will share desktop too. That's my daughter. She was 7 months old there. Um, this is a website, and I'll put this in the link below. I'm sharing. Okay, I'm sharing. Sorry about that. I'm sharing. I think it's this one. This, let's go, let's do this. So this is the lab website, fieldslab.ucsd.edu, and it talks a little bit about what we do, shows some pictures of astrocytes and neurons, pretty images here. These are from brain tissues, human brain tissues. Astrocytes are my favorite cells, what I got my PhD on, studying how these guys work and how they are altered during brain diseases. And the neurons, which you can see right here, these big long processes and these cell bodies, they are sort of the communicator or the main actors in the brain. I don't really like to say main actors because I think astrocytes are super important, but neurons have always traditionally gotten the most attention. And anyway, I don't want to get too far into that, but this, you can find a little bit about what we do, get some pretty pictures. You can look at the funding that we have, and all this is publicly available. You can read about that. You can look up any of these grants by this grant number here. So each of them have a grant number, and this is how we get money. It's all taxpayer money. These are publications, and this is not perfectly up to date. We do have some 2025 publications that are not in here yet. This is UCSD. Then you can kind of meet our team. This is me trying to be cool on the mountain. These are postdocs. Jackie is actually not in the lab anymore. These are my excellent SRAs. They are both awesome. She'll be going to medical school. Anna will be going to medical school soon. Mary wants to go get a master's or a PhD and keep on doing science. These are undergrads that work with me. Matthew should have a picture here. And then we have one more, Jack Melcher, who's not here. But they're all great. And then people who worked with us in the past. And then this is the Scripps Pier, just down the road, right off the beach. And yeah, this is where we are at UC San Diego and La Jolla. And we try to do fun stuff together, go out and eat. We've done hikes. Yeah, this is down by in La Jolla. Previous lab members doing a hike here, doing some science, presenting our science at a poster session here at UCSD. So there's that. And then we have, I wanted to tell you our main project is an R01 grant that it started in 2021, it ends in 2026. R01s are the main grants that fund labs in universities. They usually last 5 years and with an option of being extended. And yeah, it's very difficult to get one. I think probably the average success rate is like 10%. But the truth is, everybody who starts out, or 99% of people who start out trying to get an R1 are really bad at writing grants at first. And it took me like 15 grant submissions to get my first R01. And it takes a year or two just to write your first one in a way that will be competitive. So it ends up being a lot of iterations of a document that you submit to the NIH, the National Institutes of Health, and then other scientists review it in what is called study sections, and they score them. And then you get the feedback. And if it's not funded, you get to respond to the feedback one time and resubmit it. And then they will score it again, and hopefully you'll get funded. If not, you've got to start with a brand new submission. So this grant, we were looking at immunometabolic mechanisms underlying HIV-associated neurocognitive disorders. And neurocognitive disorders are just anything like you can't remember where you're going when you left the house. You have a hard time remembering your address or what's going on or words, all the way to dementia, where you don't know where you are half the time and you have these episodes. Similar in some ways to like more common dementias like Alzheimer's disease. And the reason that HIV remains a problem and necessary is because the people that have HIV, they're living much longer now. because the antiretroviral drugs keep the virus down. Most people can think about Magic Johnson. He got the disease or infected way back in the early 90s, and he's still doing well. Not all people are that lucky as far as being able to thrive. Sometimes when they get HIV, even if they have the drugs, the antiretrovirals, they have problems in the brain. And one thing about HIV that interested me is that it, the mechanisms or the pathology that underlies these cognitive problems or these problems in thinking and daily functioning, they have a lot of the same mechanisms or properties with other dementias for which we don't know what started them. Like, for example, Alzheimer's disease. We don't know what started the disease process. Like in Alzheimer's disease, you hear a lot about Alzheimer's disease being familial, but really only something like 3 to 5% of Alzheimer's cases are attributed to some gene that was passed down through the family. Most of them are, we don't know what caused them, and that's why it's such a problem. There's no strong genetic association with them. So there's a big question as to what's causing Alzheimer's and how do you treat it and how do you detect it? Well, there is a question, or there's some evidence that viral infections can predispose people to Alzheimer's disease or other neurodegenerative diseases. And HIV may be one of those, or it may induce Alzheimer's-like pathologies in some people. But the fact is, for a long time, people with HIV didn't live long enough to get Alzheimer's disease. Now they are living long enough. And so we're investigating a lot of what HIV triggers in the brain regarding mitochondrial function, metabolism. Think about how your brain breaks down food, how your body breaks down food into molecules that can then be metabolized for energy to be used by your muscles, your whole body, but also your brain. Your brain is energy dependent, highly energy dependent. It's something like it weighs like something like 3% of your body mass, but it uses 20% of the energy. I heard that a long time ago. I don't know exactly how true it is, but the point is that it uses a lot more energy per mass than most parts of your body. And that's because there's lots of neurons firing in your brain to store memories, to learn new things, to recall memories, and just perform tasks. So we're very interested in how these the metabolism, the breaking down of energy-rich molecules to keep your brain functioning, how that is affected during neurocognitive and neurodegenerative diseases. And HIV is in some ways a model case to figure that out, because again, we know that HIV infection begins an inflammatory or initiates an inflammatory cascade that ends up, in many cases, causing mitochondrial dysfunction, and that is associated with neurocognitive impairment. So how do we study this in our brain? I don't want to go through that. I thought I could just show you this. We really use 3 methods when I'm generalizing to try to figure out what's going on. We start looking at post-mortem brain tissue. So we have people who donate their brains after they die, and we can look in them and look for changes in mitochondria and inflammation and neurons and astrocytes and all the inner workings of the brain, and see how their state at that snapshot of time, and compare that to all the clinical data that we got from these people during life. And you can take a set of brains, 20 brains from people that had some kind of cognitive impairment, and compare that to brains of people that had no cognitive impairment. And if you find changes in the brains from people with cognitive impairment, you can at least say there's a correlation, there's a relationship between those changes and the cognitive impairment. And you can look at a lot of other life history to figure out what may have caused that, um, cause those changes. And then you can use animal models or cellular models, basically lab rats, lab mice, or cells that grow in a dish to try to model these changes and see if it causes problems um, similar to what you found in the brains. And then if you do, you can test drugs or therapies in these lab animals and in the cell models. And then ultimately, that'll lead to hopefully therapeutic strategies in clinical trials. So um, that's kind of how what we do. And then one of our major findings, something that I don't know how major it is, but it's major for us. It's what we've done. Um, is this, hopefully this kind of summarizes it here. We published this in 2019. Um, but what you can see here is on the left side, you have a neuron, and these little red dots are representing mitochondria. You have microglia, which are like the police officers of the cell. They kind of the brain. Sorry, they're a cell that is in the brain, and it's kind of making sure everything is all right. It looks for problems like inflammation or invading pathogens like viruses or bacteria or something that is cancer that could be in the brain. And if it finds it, it alerts other cells. These are astrocytes, these yellow cells. They are, my PhD mentor called them the mother of the brain. And they wrap around the blood vessels in the brain, but they also wrap around neurons. They provide the energy molecules for the neurons. They provide the signaling molecules, or at least the building blocks of signaling molecules for neurons. And in a healthy brain, that's what they're doing. And the microglia are just kind of waiting for something bad to happen, and the neurons are functioning as they should. In an HIV positive brain, which is represented here, the microglia, these orange guys, are active and they are reacting to the HIV and to damage in the brain, and they are sending out molecules telling the astrocytes to do different things. The astrocytes then require more energy, so they send out less of these yellow circles, which are the energy products. Instead, they have to keep them and they use them for themselves. And we think that causes the neurons not to use as much energy. And if that's happening short term, it's probably fine. And the microglia can fix the problem, get rid of it. The astrocytes can help stave off any issues, and then they'll return back to this healthy brain. But in the case of HIV, you can't cure HIV, or at least we don't know how to in most people. There are a few cases of it being cured, but most people it'll never be cured with the current state of medical treatment. So the HIV is always there, and so we think that you have this chronic or long-lasting inflammation, which is ends up activating the astrocytes, and it keeps the neurons from ever being able to go back to their homeostasis or their basal level to where they're just functioning normally. And so what we're trying to do is find molecules that calm these things down, sort of block the signal that HIV is sending, or blocks the damage that HIV is causing, even though we can't get rid of the HIV. And this could be compared to other neurodegenerative diseases like Alzheimer's disease, or there are a lot of brain diseases or dysfunction that are associated with obesity, or other kinds of chronic illness. And the point is that if you have like this chronic, whether it be obesity or sedentary lifestyle or some other genetic abnormality that is causing inflammation or some dysfunction in the brain, and in the microglia to these cells here to be active the whole time, or chronically, it could pull energy from the neurons, the cells that need it, and keep these other cells active. And so what we're trying to do is figure out how to target the brain using metabolic strategies. That could be by changing your food, by exercising more, by eating at different times. We have studies on caloric restriction, for example, eating during only certain hours of the day. And that allows it kind of shifts the way your brain gets energy, the molecules that it uses in a way that may be therapeutic. So I could talk more about that later if you find it interesting, but that's the main idea with our lab. We have quite a few different studies that are looking at these metabolic processes in disease states from different angles with different cell types, but astrocytes are probably my favorite cell to look at, although we're more and more looking at macrophages. Um, so I think that's all I will say today. Next time I might tell you a little bit more about me and how I got into science. Uh, but that's our lab. That's what we do. I'll try to do this more often. Thank you guys a bunch.